Welcome to Research Digest podcast. I'm your host, and today we'll dive into the fascinating world of immune system. More specifically, how it knows when to stop fighting. We know that T cells and B cells are often called the soldiers of our body, and they rush in to fight infections when we are attacked by something external or when there are some objects that are identified as enemy inside our body. Now, when T cells don't stop, the body begins to attack itself, leading to autoimmune diseases. So we'll uh, talk more about autoimmune diseases and how to control our immune system. So to discuss all these things, joining us today is the lead author of a groundbreaking study published last year in the prestigious journal Immunity. Let's welcome Dr. Jubayer Rahman from the National Institutes of Health. And the title of the published article is a CD4 plus T cell intrinsic complement C5AR prostacyclin IL1R2 axis orchestrates stage 1 cell contraction. Welcome, Dr. Rahman.
Thank you very much, Banna. It is my immense pleasure to be in this program. And first of all, I really like to thank you for bringing up this very important topic in the field of immunology.
Yeah, thank you
So,
for joining us today. Yeah,
sure.
before we dive into science, please share your career journey with us briefly.
Yes, of course. So basically, I started my science, as far as I remember, back in 2000 time, year of the 2000 when I was in Bangladesh. And then actually I graduated from biochemistry department, Dhaka University. That is the time I started thinking about doing science. And then I thought how to, build up career on that particular direction. So one of the aspects was thinking either building career in the pharmaceutical science during that time I'm talking about, or going to one of the renowned, research center in Bangladesh, which is called ICDDRV. Some other, along with some other institutes also. So I went there and then started doing my work, master's thesis, and then from there I started thinking back to teaching position. So I was so lucky that went back to the same department, Dhaka University biochemistry department, and joined as a lecturer. And this is something also very interesting experience because teaching was the first time in that moment when I joined this department, because I never had any teaching experience. So it was too much stress, at the same time too much to do. So I even did not recognize when the 1 year after joining was gone, like flew away. And then I started looking for my PhD position because, this is where the faculty people or science people normally, looking forward. Having a couple of offers, and then I started actually not a PhD position because of family reason. I moved to Sweden, where I started doing some advanced coursework. So that was my kind of gateway to, went to university outside Bangladesh, and then started looking for a doctoral position.
What was it the part of your PhD program or?
Actually, not. It was not a part of PhD program. So, but it was a kind of challenge because of the family reason. I had to, take that path. But when I showed my interest to my immediate, supervisor there, and it was not that easy actually. That time was really difficult for me. But finally, Allah helped me in this case that I was offered for a full PhD program.
Okay.
So the work was basically on that time infectious diseases. Mostly I was focusing on tuberculosis vaccine development. So that work ended up with a couple of papers published, manuscript, and then I successfully defended my PhD and went back to department for few months. I'm in
At
Bangladesh,
Dhaka
and
University.
then actually for many other reasons, which is not really relevant in this particular program, but
Mm-hmm.
I was not able to really continue, so I had to come back to Sweden, and then we decided, okay, maybe we should really move on to different countries. So then we decided together the family we should maybe go back to USA. And, this is one of the routes everybody almost from our background they choose whatever they're doing a PhD coming to US
Mm-hmm.
just for as a postdoctoral position and then looking for moving on to next level. So I did the same thing. I ended up in one collaborative project with Scripts in San Diego, and then I did a short postdoc there for 2 years. And actually, I was like also having two papers out of that time point. And then I actually moved to National Institute of Health in NIH back in 2014.
Okay.
The work actually I switched from my infectious immunology to more in the autoimmune diseases. So that was the
Mm-hmm.
transition actually I chose because what I learned from my PhD training that whatever you do during the PhD program, if it's possible, you can actually change the subject a little bit. Not necessarily your core, but if you
Mm-hmm.
really move on a different field, you can actually learn more.
All right. Mm.
So the postdoc period is very crucial. It is short. It is even harder than the PhD actually. A lot of people they don't understand the importance of the postdoc period because many people they did not know how important it is, and we
Mm.
also did not know that. It's because PhD is such a intense, then we get exhausted, and then we move on to the next postdoc, and then we feel okay, I am now kind of relaxing. But actually, it is totally opposite. It is even harder because that's the moment when you build your next career, like next phase.
Mm.
So this is why it's such an important phase. Anyway, so
Right.
I actually ended up then in NIDDK in NIH, where I started continuing type 1 diabetes, the field where I was thinking to do. And autoimmune field particularly, actually, I really liked it. When I moved away from an infectious field, I started thinking that autoimmune field is over fascinating because how this own immune system actually playing different
Mm.
roles during this kind of autoimmune diseases. Because someone, but that someone will be working against you. Can you imagine how difficult the situation is?
Right.
So it is exactly the same thing is
Mm.
happening in the immune system. So I started working on the type 1 diabetes and then dissecting, some of these immune cells, how they contribute to that. I was not bit lucky enough in that particular moment because, you know, there are things are not always going easily to everyone. Sometimes it's really hard, and that was one of the moments when my PI, which is actually very well recognized in the field, she worked with a Nobel laureate as a postdoc in Rockefeller and graduated from Stanford, uh, received the presidential award. Even all those things, she was not able to continue in NIH, so she left. So
Okay.
basically, she left Amgen in San Francisco. So I was not able to really follow her up because I just moved from California to Maryland, and then going back to San Francisco with her was not that an easy decision. So
Right.
then I thought, okay, no, I have to really find in a quickly different, lab. Then I was contacted by my current lab where actually I started working with, and Dr. Claudia Kemper. She actually moved from King's College that time to NIH. So she's one of the really pioneer, you know, scientists in this field where I started
Mm.
working even deeper, understanding how these autoimmune diseases, in relation with some complement molecules. So just for the audience, the complement is is a very ancient molecule. Like a lot of many years ago, people already recognized the complement protein.
Right.
Um, it is in a simple manner how people know it. When you face an infectious pathogens, So this complement can, just opsonize it, and it's mostly from the liver derived complement.
Right, right.
So it's like in a systemic environment, opsonize it and giving it to this own immune cells to destroy it. So this is
Right.
how it works. But
Yeah,
then,
that's really interesting. Yeah.
yeah,
Yeah,
exactly.
we'll learn more about the complement systems. But
Yes, exactly.
as you're saying, your broad research focus is the mechanisms behind autoimmune diseases, right? So yeah, just tell us more about autoimmune diseases. What are these diseases, and give us some familiar examples so that
Sure.
we can relate
So
to.
as I started talking about the type 1 diabetics, which is quite interesting. There are two forms of type 1 diabetics, like type 1 and then type 2. So
Mm-hmm.
type 2 is like metabolic issues. For example, someone can get it, later in life, when normally the, younger people, they don't get type 2, but type 1 is where your pancreas is not making insulin. So it is a big problem. And then, cells basically hyperactive, making, Th1 inflammatory response, and then destroy this, pancreas. This is how the person will be without any insulin, and then having all metabolic issues like glucose is up in the blood all the time, and then complications started. So when I focused on that type 1 diabetes, I really learned a lot in many aspects, that how the immune cells actually behaving. The own immune cells actually going back to the pancreas and then destroy the islets, which is the main, small microenvironment for making the insulin. So the cells can should not really attack their own thing, right? But they started attacking, and then they make the inflammatory environment, and too much inflammatory environment also not good. So why something is quite important, that the whole immune system, sometimes it can fight, sometimes it can actually stop fighting, and how they control it, this is very very important. And my current work, actually the paper that you just mentioned for this program, this is exactly what we try to understand, how the immune cells, when they should trigger the response and when they should stop the response. So that is the very critical mechanism we try to address from one angle for the CD4 T cells. And then there are many other autoimmune diseases, for instance, like rheumatoid arthritis, Crohn's disease. So some of these diseases also have various, aspects of complications, where it could be T cells, it could be some other, immune cells involved. So we try to address in this paper a couple of other diseases also. You probably noticed that we mentioned Crohn's, we mentioned rheumatoid arthritis, we even mentioned the CAPS disease, which is like an acquired inflammatory response disease. Some people can have genetic mutation, and it's a gain of function of inflammation, like cannot control the
Right.
body, so it's always on the inflammation. So yeah, so those are the couple of autoimmune diseases we try to really address, how our findings from the basic research can translate. So that was the beauty of that paper.
Okay. Thank you for sharing. So, you here in this particular paper, you discovered uh, axis of a axis of, let's say, the action mechanism that kind of explain how T cell contraction takes place. So, briefly tell us why this contraction phase of T cell is as important as like the activation phase.
Right. So this is really, really good question, Banna, because when you started back 15, 20 years ago, we didn't really learn a lot of things in the immunology when it comes to the lymphocytes, like T cells, B cells. We just know that they can do something, but we never actually thought about that they can also control themselves. So that is the very interesting part. Second part is also very important. When they control it, they can be controlled by their metabolic status. So you see how it's connected. So in the recent findings, there are a lot of things connected, that what we eat, that's also contribute to their, the immune cell function. We never even realized that thing. So now coming back to your the core question, how we think about this CD4 T cells, particularly in this paper. So the T cells activity is an adaptive immune system. Basically, two types of immune system we know.
Hmm.
One is the innate, which is really a fast responder, and they don't have the memory. So basically, they come and check and then do the job, and they
Activate T
forget.
cells and everything.
But then there is another phase is coming later. It is the adaptive immune system where the T cells, B cells are very important, but they can have their memory. So meaning these T cells has to be more sustained in the system in different format because of that memory. If they do the job and they die, that means you're losing those cells. So meaning there are some T cells could be effector function, which means they're the main player, and there are some cells there could be some like naive form, so they're not really doing anything but maybe in the system, so probably they're just ready for the next call, something like that.
Okay.
So the effector cells, when they're triggered by something, they make some strong cytokines are basically the key molecules where they can go for function, if I say in that way.
Okay.
So when they make the cytokines, for instance, if I name one of them is the interferon gamma,
Hmm.
among many others, it's a true signature cytokine for Th1 cells. So when they're making it, they need to be controlled at some point, because if it is they keep making, it's not good for them. It's really energy burden, so they need to, if you go back to the metabolic phase, you need to use a lot of energy in that way too. And also too much that inflammatory environment not good for many things, including the cells also. So what we found in this paper very interesting, that the cells that are making, at some point they're switched to IL-10, which is another cytokine, which is not an inflammatory cytokine, which is like a negative regulator, if I say in a simple way. So basically, it just is a working as a, like a brake. So for example, you started driving the car, making the speed 60, 70 miles per hour, but then all of a sudden, you see a police car, then what do you do? You just brake. So you still, car is moving, but you coming back to lower speed, right? So making cells for IL-10 is doing exactly the same thing. So it's a breaking point, and the cells become steady, they survive. So we found that the cells goes two phases, from effector function to double cytokine producing cells, both interferon gamma and the IL-10, and they become stable. So they are not dying, they are surviving better. And then in this paper, we try to understand what actually behind this thing, how this is happening. And we found very interestingly, when it comes to the metabolic phase, like the prostaglandin, prostacyclin, complement receptor, is an axis how they build and work function together. So in this particular paper, we focus on the complement protein C5, which is working with the C5 receptor 2, and then it engaged with prostacyclin, which is coming from the arachidonic acid metabolic pathway, and then it works with the one of the inflammatory cytokine receptor, and the IL-1 interleukin 1, which is a very critical molecule for early phase of inflammation. This is also the very beauty, if you look for how IL-1 can be controlled apart from T cells. So IL-1, when it's increased, there's a receptor, it's called IL-1 receptor 2,
Okay.
IL-1R2. That receptor neutralizes the function of inflammatory cytokine IL-1.
Okay.
So you see that how the cells are basically making it, but they're also making something to neutralize it. So it's always a kind of a nice balance they maintain. So we found that this complement receptor, IL-1 or 2, and the prostacyclin, this is an axis. This axis actually play a very important role to have a nice and function of CD4 T cells. Now,
Okay.
question is, if this balance is disturbed, like changed for some reason, that's where the problem comes. Either it's too much inflammation, or actually cells are not responding when they need to respond. And this is where we found some of these complications like Crohn's disease, rheumatoid arthritis, CAPS patients, we found that this axis is disturbed. It's not really, it's dysregulated somehow.
Mm.
So we found that, okay, so maybe this is very important to have these axes functioning properly so that, the complications never happens.
Okay. Okay.
So that was the very fundamental finding and common pathway in different autoimmune diseases we found.
That's wonderful. Okay, you mentioned complement C5. So when we think about complement system, we think them as a soluble proteins floating in blood, right? But the specific C5 receptor A receptor 2 that you studied in this particular paper, they are inside the T cells. So how does this intracellular version of complement work, and how does this kind of change our
Yeah.
understanding of T cell
Right.
function?
Yeah, this is another excellent question. So, I mean, even I'm pretty sure even though you're not in the field, but your question is very to the point. So, as I told you in the beginning, that the complement comes from systemic environment mostly, like the liver derived, which is the concept for many years.
Hmm.
But then our group, including some other group, they actually found that it's an intracellular complement that's shaping the whole immune cell function. So
Yeah.
in this case, it is actually not dependent on that systemic complement. So
Hmm.
meaning the cells we are currently working with, like either T cells or monocytes or macrophage or CD4 T cells, even B cells, we found that this complement, they are making it. The cells are actually making it intracellularly, and they do have those receptor on their surface or intracellularly. So the receptors, depending on which receptors we're talking about, it could be on the surface or it could be in the inside the cells. So whatever they're making, it can be released. So it's a kind of autocrine function. It can be released and then it can be bound with the same receptor and giving the signals downstream. So we have been actually doing a lot of cool stuff, and we found that a lot of nice things are happening inside the cells, so which is out of context in this paper, but it is actually not just immune cells, even non-immune cells, they are also can making complement. So the
Oh,
whole concept of complement field is totally changed, and it's connecting beautifully with the metabolism. So that is really important thing.
that just reminds me that we don't know I, a lot of things yet.
Exactly. So this is what just you mentioned, that this is the beauty of the biology. So there are things happening. We're just trying to understand. So we don't need to really make anything new. We need to figure it out what is happening and why is happening. So those are the questions for us, like field in the biology. It's very different from, you know, like other people working in the computer science or something. They need to make completely new thing. But in our case, we just need to figure it out. There are millions of things happening. We just need to know what is happening, is it new, and why is happening.
Hmm. Okay.
Yeah.
Yeah, that's great. So I have one more slide to kind of briefly summarize your particular paper, like the
Oh
major
yes, sure.
findings. Yeah, if
Right.
you can
It'd be nice
kindly
if you can bring it
explain.
up.
Yeah.
Yeah, sure. So yeah, from this paper actually, as I mentioned, it's really a complex work. I nearly did not mention how many years we invested for this paper during the COVID time, and then there are so many collaborators involved. Publishing paper is really tough job. It's really tough job nowadays. You need a lot of resources. You need collaboration. So this paper is an ideal example how many years time and the effort and also the ideas all came together and then ended up with a nice story. So as I just mentioned that the main work was on the focus on the CD4 T cells, and then we try to understand how these complement receptor, in this case the C5 receptor 2, and I was talking about that, how this COX2. I didn't mention the COX2. If people are in the field familiar, this is one of the enzymes that actually work on arachidonic acid.
Hmm.
this pathway from COX2 that works on the arachidonic acid, making the prostaglandin. It is very known, and then prostacyclin. So those central pathway, which is basically downstream of this C5 receptor, which we found, is extremely valuable because when you feel pain, what do you do normally? We just have some painkiller, right? So for
Yeah.
example, aspirin or some other ibuprofen. So what it does is actually block this prostaglandin synthesis, and then you feel better. But we never actually thought this blocking of this thing, how this little change can change many things downstream, because we did not really connect it, right? So now when we started working on CD4 T cells, we found that there are a lot of blockade molecules we used, like COX2 inhibitor, prostaglandin inhibitor, prostacyclin inhibitor, complement receptor inhibitor, and we try to find out downstream changes also. The reason we were so much interested on that, because aspirin has been giving for people, because when it's a lot of people taking in the heart disease complications, the reason is too much prostaglandin and then the thromboxane, which is another molecule causing the blood vessel shrink. On the other hand,
Okay.
prostacyclin, which is a vasodilator, so it's making the blood flow better. So you see how this pathway is even regulated. So this is why they are blocking this thing, so
Mm-hmm.
that you do not have the thromboxanes downstream. So we actually try to address this in this pathway, how prostacyclin versus prostaglandin, these two are regulated downstream of complement receptor, and how these things are changed when there's some disease condition like autoimmune diseases. So as I said, when it comes to the autoimmune field, this pathway on the left side, it goes up. When in normal situation, these are well balanced, but in order to inhibit this autoimmune reaction, you do need some kind of inhibitors on the left side of this pathway, so that you can re-establish the harmony of this pathway by
Mm.
making some prostacyclin, so that it doesn't get imbalanced. And this is exactly what we have drawn it here. So allowing the cells, making interferon, allowing the cells IL-10. So if one is higher, one is lower, that would be the problem. And then actually we found that very interestingly here, how the Th1 cells induction, which I said it before, they go to the Th1 phase when they are contracting, they have a reduced Th1 response. And to that end, prostaglandin, prostacyclin, they are the really central mediator, and complement actually in the upstream of those. Yeah,
Right.
it was a good piece of work actually. At the end, we were able to understand this thing.
Right. Yeah. Thank you for sharing. And
Sure.
you observe that in several autoimmune diseases, when you disrupt this pathway, this axis, and the T helper cell persistently increase or persistently stays in the
Getting
system.
hyperactive. Yeah.
Hmm.
They got
Okay. So
hyperactive
that that's
under the
very
inflammation.
relevant to our diseases, like some of the most important autoimmune diseases. And now, is there any future perspective from your study that will have in future a drug that will kind of shut down or this abnormal abnormal persistence of the T cells and
Right.
kind of maintain the balance?
Right. So yes, that's actually the goal of that study.
Mm.
We try to really find out any targets uh, we can identify, and then we can use some kind of drug or something. So as I mentioned, this prostanoid pathway, which is
Mm-hmm.
kind of very old pathway. So there are a lot of molecules people have tried.
Okay.
What people haven't tried is that which inhibitor and what doses. These are very important. For example, you can actually block the pathway completely, but then actually you are not helping. So you
Mm-hmm.
have to do certain dose that can maintain the balance of those downstream,
Mm-hmm.
all the molecules. So that is something we found. And we found that the COX2 inhibitor, as I said, that was our first target because people already used it. If you use the COX2 inhibitor, that downstream everything you just blocked.
Hmm.
but then we use the inhibitor moderately, like not full block, like partial. And when we did that, we actually re-established the whole pathway nicely and reduced the burden. So same way, when we go went downstream of COX-2, there was another one is the prostaglandin synthase enzyme, which is responsible for making prostaglandin. So we wanted to block that one also because as I said, prostaglandin is a kind of inducing pain, inducing hyper TH1 activity. So we also use the block it for that particular pathway, and we found that this one also works really good.
Hmm.
So another important molecule that people already knew but did not know in the context, which was the IL-1 receptor 2, which is a neutralizing molecule. Like you have the more inflammation, but you do need to suppress it. And then IL-1, which is can be found also in a soluble form,
Okay.
not both in the membrane bound and soluble. So that given that also reduce the inflammatory burden. So yeah, exactly. So there are a couple of targets in that pathway which can be used not only even the T cells. I believe that some other immune cells, those are also involved in this kind of complexity. They might have the similar con- context, like imbalanced of those pathway, and perhaps the magnitude of this response could be even higher, especially when it comes to the innate cells, because they really make a lot of key cytokines. T cells obviously can make even less, but some could be of course more like interferon gamma. So I think that the findings of this paper can actually extend it to those other cells, even the non-immune cells also. So, but there are some studies needed to find out where it
Hmm.
can fit much better this whole access.
Okay. That's great. Thank you for sharing your insights and simplifying this wonderful paper. And we are almost at the end of our conversation. So finally, I just want to ask you if you have, please share if you have any last minute suggestion for our audience. It can be about science or it can be even about diet, because we are talking about diet is
Yeah,
very much
right.
related to
Exactly.
our health and the diseases.
I think if I pick, okay, let me pick both because
Mhm.
I
Sure,
mean,
sure.
science is important and the diet also important. One
Mm.
is dependent on another thing because our life is also dependent on the diet. So, regarding the science, since we work in the scientific community, uh, I think the challenges are really huge. Science progressed a lot, and that depends on many things. As I mentioned, in my this project, we actually involved two international collaborators, three local collaborators. So there
Mm.
are a lot of efforts, and then of course, in terms of investment and money, it was also huge. So when it comes to the science, I think it is very important to develop the good hypothesis first of all, what questions you are asking, and then try to think that how much actually is known and which direction it can bring you the
Mm.
new findings. So assessment of that thing is quite important for every scientist. Of course, I mean everybody can publish paper, we can publish paper, but the thing is that how much deeper you can go and what can actually have some kind of impact on human health and disease. So that direction requires combination of many things, not just one or two things. So one suggestion could be that selecting a project and then building the concept around that question is quite important.
Mm.
And the second thing is, as I always love to understand now, since it's a metabolic things is quite important, especially there are a lot of metabolic diseases, and obviously the recent trend of understanding the biology is truly dependent on how cell metabolism is changing, because this is contributing to the cellular function. even the both immune and non-immune cells. So meaning whatever we are choosing to eat, that can also change the cellular function, even your immune cells, your body, other cells. So that being said, there are a lot of papers published, especially um, in the recent time when it comes to the metabolic diseases. That the truth is that, I mean, fasting, keto diet, all those things really contributing to how immune cells functioning. Your immune cells can be reboosted somehow, depending on the diet, depending on the duration of fasting.
Wow.
So people normally love to say there are not a lot of findings how these metabolic diseases could be controlled without medication. But actually, it is not true. It is the lifestyle, it is the diet, how you do exercise, how you maintain your routine, whether you are stress free or not, how much time
Mm.
you are happy, how much time you are sad, how much you are distressed. All those things are really in combined form. But when it comes to the medicine, the medicine can target only one thing. If somebody says I cannot sleep, they can give you a sleeping pill. Somebody say, okay, I have glucose high, they can give you some medication to go become glucose gets lower. But as you say that your number of medication is increasing a lot at the end. But as soon as you stop the medication, you having the problem back again. So maintaining the lifestyle and do work as much as you can, and then having your diet properly selected is quite important. So that's all I can say, at least from now,
Uh,
whatever we have been doing
sure,
so
sure.
far.
Thank you so much. A very important message. Uh, Thank you so much for coming to this platform and for your time. And thank you, everyone, for watching us and listening to us.
Yeah, thank you to the audience, and thank you very much for organizing this kind of program. And I really wish you the best, successful, more nice article presentation through this and your program. Looking forward. Thank you, Banna.
Thank you so much.
Sure.